So we're about to start chapter 16, so we're going to start talking about host-microbe interactions. So we have to remember that we actually contact bacteria constantly, right? We breathe them in, they are mixed in with the food we eat, or what we drink. We're constantly touching them, and a lot of them do not harm us. I've already grossed you guys out with some of the stuff that lives in and on our bodies. Now, they can colonize body surfaces. Others we actually will shed on a regular basis as we shed cells. A lot of the things we swallow will actually die in our stomach or get eliminated with our waste products. The ones that do cause damage are the ones that we call pathogens. And remember, I keep telling you not to focus on the pathogens because they're such a small portion of microorganisms. But we are going to start talking about them in a little bit more detail now. When you look at pathogens, they're going to have certain characteristics that allow them to be pathogenic. So remember, most microbes are actually harmless, and they're part of our normal microbiota. So they normally reside in or on our body, and the relationship is delicate. So things can actually throw off homeostasis, and this can allow certain species to become opportunistic. Also, if you are an immune-compromised individual, there are organisms that are normally in a healthy individual's microbiota that don't make them sick. But if you're immune-compromised, they can actually cause disease in a patient. Now, I mentioned this the last time I saw you guys, but we have symbiotic relationships. Remember, when you have a symbiotic relationship, you have two organisms living in close association with each other. In a mutualistic relationship, both organisms benefit. In a commensal relationship, one benefits and there is no effect on the other. And in parasitism, one benefits and the other is harmed. Now, your book sort of indicates commensalism happens. I can tell you from a scientific, non-medical perspective that's not really true. Most organisms tend to either be mutualistic or parasitic. Now, when we're looking at our microbiota, we have organisms that live on us for extended period of times, but also for temporary periods. So resident microbiota will actually live in or on you for an extended period of time. Transient microbiota live on us or in us for short periods of time and then leave. We have a feeling that these may be rather important for human health, but we don't really know a lot about them. And there is actually a human microbiome project in progress to look at our microbiome to try to understand it better, especially using metagenomics. So we want to actually look at DNA sequences to find out who's there. So comparing those core genomes. And part of it, remember, is we have trouble culturing a lot of things. So we need to figure out who's there using methods that don't require culturing. All of us are colonized by our microbiota at birth. And if you're breastfed, there is a contribution from breast milk to include with your microbiota. However, as we get older, our life experiences cause us to have different compositions over time, and the changes occur due to the physiology and lifestyle of the host. Now, dysbiosis is when you actually have an imbalance in the microbiome, and this is related to some disease states. I've mentioned before that there are some patients with Crohn's disease where they've had a fecal transplant, and suddenly they no longer have symptoms of Crohn's. So is Crohn's really an autoimmune disorder, or are we dealing with this imbalance of the gut microbiota that could be treatable? And your normal microbiota are really important in helping to protect against pathogens. Sometimes they will cover binding sites of your cells to prevent attachment of parasites. They will consume available nutrients before parasites can. And they produce compounds that are toxic to pathogenic bacteria. However, if you suppress them or kill them, this can allow pathogens to colonize. Part of what allows C. diff to take over when you're on antibiotics is the antibiotics are killing the healthy bacteria not just the bad ones. Now, with your microbiota, they do help stimulate the adaptive immune system. So antibodies that will form against your normal microbiota can also bind to pathogens. And you find that when they've done experiments with mice that have been raised in microbe-free environments, they have underdeveloped immune systems. This also seems to be very important with oral tolerance. Your immune system stops responding to microbes that are in your gut and your food. So there's this idea of the hygiene hypothesis, right? That some of our allergies and those with weaker immune systems, it might be because they did not come into enough contact with microbes when they were growing up and developing their immune system. So they don't have the appropriate reactions to microbes anymore. And if you don't come in contact with certain compounds growing up, you can develop allergies to them. So colonization actually refers to the microbe establishing itself and multiplying. When you look at the word infection, we're talking about pathogens specifically. We tend not to use the word infection unless it is a pathogenic species. And infections can be subclinical, where you have no symptoms or you may have mild symptoms. When you have infectious diseases, you do have a noticeable impairment. Symptoms are going to be subjective effects experienced by the patient, things like pain, nausea. Signs are going to be objective evidence. There is or is not a rash. There is or is not swelling. There is or is not pus. When you look at the initial infection, we call this a primary infection. Primary infections can lead to secondary infections. So there's damage caused by the primary infection that then leads you to be susceptible to these secondary infections. A good example is when you have a respiratory illness, it damages mucociliary clearance, which then can allow other bacteria to cause a second infection in your respiratory system. Now, when we're talking about pathogenicity, primary pathogens are going to be microbes or viruses that cause disease in otherwise healthy individuals. Plague, malaria, measles. You get it, you get sick. When you talk about opportunistic pathogens, they only cause disease when there is some sort of compromise to the body's immune system or when they're introduced somewhere they're not supposed to be. So, for example, Pseudomonas is part of the normal microbiota of some people, but you can get Pseudomonas infections. A lot of women have Candida in their vagina. However, when you throw off homeostasis of that environment, you can end up with a yeast infection. Now, the word virulence actually refers to the degree of pathogenicity, and virulence factors allow a microorganism to cause disease. So when you're talking about communicable and contagious diseases, these are ones that are easily spread. The infectious dose is the number of microbes necessary to establish an infection. So if you look at something like shigellosis, you need 10 to 100 cells to actually be at risk of infection. When you look at salmonellosis, you might have 1 million cells that have to be ingested to get the disease. The infectious dose sometimes is reflective of how good a pathogen is at surviving stomach acid or getting past the body's defenses. Now, the incubation period is the time between the infection and the onset of symptoms. This can be a few days. It can be years for something like leprosy. It really depends on the growth rate of the organism, how healthy the host is, and the infectious dose. Illness are signs and symptoms of the disease, and you may have a prodromal phase where you have vague symptoms. Basically, you feel like crap, right? You have malaise, maybe a slight fever, you feel smushy, stuff like that. Convalescence is the recuperation period where you are recovering from the disease. And if you're a carrier, you can actually harbor, so you have or you spread the infectious agent for long periods of time, even though you don't have symptoms. Now, when you're talking about different types of infections, you can have acute, chronic, and latent infections. Acute infections are when you have symptoms that develop quickly and they last a short time. Strep throat, right? You have the symptoms, you feel better after a few days on antibiotics. Chronic infections will develop slowly and they can last for months or years. It can take years to develop symptoms for tuberculosis, and then even on antibiotics, it can take months to years to get rid of it. With latent infections, the microbe is never completely eliminated, and it will continue to live in your tissues without causing symptoms. So, for example, chickenpox never goes away, and because it's hiding in the nervous system, it can return as shingles later in life.